Very excited that we're able to be here in person and streaming on both Zoom and YouTube. So it's just great to see everyone. It's also great to have this talk. As you guys remember, we had a search for a numerical oceanographer or computational oceanographer last year, and we got to get an introduction to the type of stuff I think we're going to hear a little bit about today and how the relevance of our new faculty position. So I think Dr. Edwards' presence today is incredibly timely for the types of collaborations we can be doing across the pond between Moss Landing and UC Santa Cruz. So Dr. Chris Edwards is a professor at UC Santa Cruz. He's in the Ocean Science Department. He did his bachelor's at, Haverford College. I knew there was going to be a noun pronounced differently on my end. He did his PhD at MIT and Hui in 1997 and ended up, after a little bit of time in Berkeley, at UC Santa Cruz in 2002, which is when I arrived at Moss Landing Marine Lab. So we are both celebrating our 20th years this year, so really exciting. And this is the first time I've met him, so it was fantastic getting to talk a little bit with Chris. You might have seen from his bio that was sent out, or if you've seen some of his other work, he's very interested in regional ocean modeling, which, again, is extremely timely right now in terms of its relevance to planetary issues that we're all facing. So I think this team and this type of work is just incredibly exciting right now. Chris's team is specifically interested in the data assimilation part of this, but I really like the fact that it's interactive and it's really trying to go across biogeochemical, physical, biological, and I think that's what you were saying some of your best interests are in, the physical biological interactions component of this, how it relates into fisheries, you know, at one point for us selfishly aquaculture, and how it can help model the types of resources that come into these systems. So with that, I wanted to introduce Chris, and the title of his talk, Modeling Studies of the California Current System. He'll go ahead and handle the questions at the end on your own, and welcome.
Thank you. It's really nice to be here, and thank you for the invitation. A couple of things. One, I'm slightly modifying my talk title. I've added the word dispersal because I'm going to be talking about dispersal modeling studies. Even though a lot of the work that I do up at Santa Cruz has to do with data assimilative coupled biogeochemical physical modeling, which is like weather forecasting of the ocean, I thought for this talk it would be more interesting to this group if I talked about larval transport and dispersal, what we can learn from looking at Lagrangian trajectories within ocean models. So I'd like to acknowledge many people who've contributed to this work. In particular, Patrick Drake, who's a technical assistant in my lab and I've worked with for many years, and a lot of the work that I'll be talking about has been collaborative with Patrick over the years. And then Anna Lowe, who is a PhD graduate from my lab from a couple of years ago. So I'll be talking about work that we all did together, and then collaborators on different parts of the work include Stephen Morgan, Mark Carr, Pete Raimondi, Hal Batchelder, Zach Powell, who was my postdoc advisor many, many years ago, and then Ed Dever as well. So I'd like to acknowledge also support through PSCO, California Sea Grant, and the National Science Foundation for different parts of this work. What I'll be talking about is you can sort of see in this figure, it represents the Central California coast, and there's an ocean circulation model. You can see the vectors that represent surface ocean currents and sea surface temperature, and the white dots in there represent particles that we put into a numerical model and track them around with the ocean currents to understand what the currents are doing for dispersal. An outline for my talk is as follows. I'm going to start with some motivation and talk about the ocean model a little bit. And then I've chosen to kind of alternate topics that have to do with physics and have to do with coupled physical biological interactions on and off. So I'll talk about connectivity matrices associated with physical circulation alone, talk about the influence of biology on that, talk a little bit about about a basic theory for cross-shore dispersion in the coastal ocean, go back to the predictability of this system and whether it is predictable, and then move on to submesoscale fronts, which is kind of a topic of active interest currently. Over on the right side is a really nice figure that I'm going to just highlight a few things now. So in talking about dispersal of propagules in the ocean, a key concept is the pelagic larval duration, which is the period of time when larvae are effectively planktonic, moved by ocean currents. And then there's some sort of competency period. After some period of time, the organisms may be biologically suitable to settle. And then the second line has to do with their different life stages, which of course influence their settlement period. But the last couple of lines have to do with their influence on their environment. So at some stages, either through buoyancy or through swimming, they can control their position within the water column. As they get to be larger, they can potentially control their horizontal position through swimming. And they can orient themselves perhaps through a number of environmental cues. So I'll be talking about all of these things on and off through the talk. And I'd like to emphasize that I'm not a biologist, and so I'm undoubtedly going to make errors in the language that I use because I'm trying to cross this bridge between physics and biology, and I do my best, but I apologize if I make embarrassing errors in certain language. Please feel free to correct me. Oh, I meant to mention, I'd be happy if people interrupted and asked questions all through it, either here or online. And so I encourage people to just ask a question if something seems out of sorts or you're curious. I'd much rather do that in the middle than at the end. All right, so motivation for larval transport studies. Well, what are larval, what do I mean by larval transport? So dispersal represents the exchange of genetic material between different populations, and dispersal in the ocean, of course, is mediated by ocean currents, and it's a really interesting multidisciplinary problem where influences of biology and physics come together to try to understand it. In a recent review paper, Swearer report, larval dispersal is arguably the most important but least understood demographic process in the sea. So this work is trying to understand something about this in our backyard, the California current. There are lots of reasons why we might study this. This middle one has a nice representation of two different loci, the genetic loci within barnacles. And the pie charts on the left show that there is a different distribution of this genetic material at different latitudes along the California current. And one of the possible influences for why there's some genetic differentiation has to do with how far ocean currents can transport a material. Of course, marine protected areas are designed to create habitats with enhanced growth. Organisms that live in the marine protected areas release larvae that then are planktonic and move with ocean currents. And trying to understand the network of MPAs, how they are locally refugia, but they supply information or supply genetic material to other parts of the coastal ocean as well as the other MPAs is also an important motivation. Are the MPAs working as a network, or are they just isolated pieces of refugia? For this work, over on the right, I'm going to focus on really near-shore populations, and that's sort of reflected in the marine protected area distribution. They're all very near-shore. And over on the right is a nice picture of Monterey Bay, which we're all very familiar with. But I'd like to emphasize that for many organisms, it's the near-shore habitat that's most important for their survival. Some are benthic-oriented, some are relying on intertidal environments, anyway, this area in red, in yellow, represents a very narrow range of nearshore environments where populations exist and have to redistribute to. So this talk is really focused on that nearshore environment. All right, the ocean model that I work with is ROMS. It's the Regional Ocean Modeling System. It's one of a handful of state-of-the-art fluid dynamics models available worldwide. It's arguably the dominant global ocean model for regions, coastal regions, certainly in this country and arguably in other locations as well. This is an example of the sea surface temperature. In our typical model, you can see the domain extends from Baja, California up to Washington State. I'm showing an animation of sea surface temperature. It's realistically forced with surface forcing from a quadruply nested atmospheric model with high resolution along the central California coast. It's forced at the lateral boundaries by realistic data simulative ocean model output. It resolves, even though I'm just showing the surface, it's resolving the full ocean depth. It's got complex subgrid scale mixing. It's spun up for a period of time. I like this animation because it shows the complexity of the California current. If you look at the movie over time, you can see that the sea surface temperature goes through a seasonal cycle. We've got winter time, which is mostly in blue, and then summertime comes in red and yellow. But even in summertime, you can see the intense upwelling that's taking place near the coast with the blue stripes and filaments that are strongest at the coast. If you move a little bit offshore, you can see the strong mesoscale variability with eddies much larger than the coastal filaments. So the way we operate is we seed these circulations with particles, and we simply integrate them using a variety of tools, but it's a very simple procedure. dx dt is equal to u. We just integrate that forward with the particles and then study how they go. The work of Anna Lowe was in a nested model domain. So the quadruply, or it's a very large sequence of nests that get us down to the central California coast, Monterey Bay, Carmel Bay, right here at very high resolution. And over on the right, I'm showing the sea surface temperature with particles that have been seeded. And even though this is kind of a slow animation, it gives an idea for what's going on. Particles in this case were released in Carmel Bay to understand how they were dispersed to other marine protected areas in the region. And you can see that the floats trace out fronts as well as end up being distributed over broader areas. All right. The first thing one does when one does an ocean model is to evaluate its behavior with respect to observations. Ocean models are very complex. They've got a huge number of grid cells, and observations in the ocean are sparse. They're sparse in space, they're sparse in time, and so there are a lot of different model data comparisons that one can do, and I'm showing like 4 here to give an idea of what we do. The upper left shows data on the left and model on the right for multi-years. It's showing an average velocity, direction, and magnitude for a 3-month springtime period. The data comes from subsurface drifters or near surface drifters, and the model shows the uppermost level for the model. What you can see is that there's some general agreement in the southward transport of the California current. The direction is indicated by the color in quadrants. So red is kind of to the southeast, black is to the southwest, green is to the northeast. And while there are some pieces of agreement, there are also some clear disagreement. If we look at other pieces of information, such as this set of figures here, This is looking at one mooring off of Bodega Bay called D90, which had a long time series there. And what we can see is the near surface alongshore velocity, both the data and model, there's high correlation with the cross-shore velocity. If we look at the average structure, the cross-shore structure is pretty well represented between the model and data. The alongshore velocity, perhaps there's a little bit of a deep bias to it. I show these not to say that the model is perfect. The model is a model. It's an imperfect representation of nature, but it has elements that we think are reasonable in trying to understand what the ocean circulation is doing. All right, so I'm going to start with Lagrangian PDFs. So these are probability density functions, and these are for floats that move three-dimensionally. And this figure over on the left is some data. These were floats that were released, I think, by Jack Barth and colleagues many years ago. But some were released near Cape Blanco, Coos Bay, and then others were released by near Point Conception down in the Santa Barbara Channel. And if we just look at the model of Lagrangian PDF, so what you do is you release floats for a long period of time, and then you create basically a two-dimensional histogram of what the density is. And in this case, the density is largest in the red location, and it decreases. It's a logarithmic scale, so it drops off rather enormously. But you get an idea from these clouds where the particles are going. In this case, all of these figures are showing releases from the Santa Barbara Channel, the same as this red patch. This is 40 days after release. The spaghetti plot kind of shows where things go. 30 days after release, we've got floats that make their way from the Santa Barbara Channel as far north as Monterey Bay. It's a little bit less so in springtime, maybe up to San Francisco Bay at low amplitude in summer and fall. Over longer periods of time, the dispersal tends to be longer, and the concentrations tend to be weaker. So anyway, the clouds get larger, the hot spots get weaker. But anyway, if you look at the, for example, northward extent, it's about the same in the red as in our model output. All right. One thing that we can do that can be helpful for understanding the dispersal is to calculate a connectivity matrix. The connectivity matrix works as follows. You divide your domain up into a series of regions. We might call them cells. And then you quantify the number of trajectories that initiate at one location and end up in another location. So in this example, I have something that's released from the green, makes its way through the ocean circulation, and ends up at site 4. I have a couple of trajectories that make their way to site 2 here, sometimes in a circuitous path and sometimes in a more direct path. And then if you do this for thousands or millions of these floats, you can calculate statistics of the distribution. And the way we present our connectivity matrix is we have the release region on the x-axis, the potential settlement region on the y-axis. So organisms that are released from region 5 end up settling or potentially settling in our model in region 4 or in region 2, and we color those according to the numbers of floats that we get. I'd like to say that in this work, we neglect a lot of biological factors that are important in nature. So there's no predation, there's no larval mortality, there's no habitat suitability. We're trying to look just at the physical circulation impacts. And this is the type of thing that one gets. So over on the upper right is our domain, and we've labeled things according to their release site. So we've got Palos Verdes, Point Conception, Point Bouchon, Monterey Bay, Gulf of the Farallones, Point Reyes, Point Arena, Cape Mendocino, Cape Blanco, blanking on that, and then the Columbia River. Oh, Hecate Bank, that's the one. And then if we look at the connectivity matrices, I'm going to start with the one on the left. This is for a winter release. So winter release might mean December through February. And we track them for, say, a 30 to 60-day window when they could potentially settle. So these have a PLD of 30 days with a competency period following that. And that's moderate. And then for some organisms, maybe a longer pelagic larval duration is more useful, maybe 4 months. And the black line here that moves diagonally represents self-recruitment. So this is cases where organisms are released from a particular cell. They go through the ocean circulation. They end up in that same cell. What you see is that for these kind of moderate PLDs, you get a lot of self-recruitment with high concentrations along that diagonal line. But there's also some trends which you can see, and those I'd like to highlight. We have a typical circulation from Southern California up to the southern part of Central California, say from the Santa Barbara Channel or from off of LA up to Point Sur. If we start up at Hecate Bank off of Oregon, we get some transport to the north in wintertime and also quite a bit of transport to the south down to Point Arena. In addition, you see some variability on here. But that's kind of the main thing I'd like to point out for this figure. Now, for a longer time period, you're starting to sample the upwelling circulation. The transport shifts in direction a little bit. You can see that the overall settlement gets a lot less than the shorter PLD. And there's quite a bit larger transport extending from off of Oregon down all the way to Central California. And there's some shift from Southern California to other regions in Southern California. The other thing I'd like to point out is the stripe of settlement or potential settlement that takes place around the Central California coast. There are kind of hotspots where multiple parts of the coast will see the particular area, and those are of interest for understanding how marine protected areas can function together. There was a question here?
Cell, is it when, so you release the particles and then they infect, right? And is it when it's in a nearshore cell or when it, like, you can tell the particle hits the shoreline?
No, it goes into a nearshore cell, which the cells are kind of shown over here on the right. This is for the Santa Barbara Channel. What we do is we divide, thank you for asking, I should have explained this. We divide the coastline up into a series of regions that are larger than our grid size. And so it's larger than the circulation. In this case, I think that's about 10 kilometers cross shore, maybe 40 kilometers, maybe 30 kilometers along shore. And then anytime a particle goes through that cell during the competency period, we treat it as a potential settler. Thanks for asking. Okay, so the first thing I'd like to point out or get back to is that these connectivity matrices depend on season. So with a springtime release, they're sampling a different seasonal structure in the ocean circulation. You see some of the same behavior, but the hotspots of settlement become a little bit more distinct with a springtime release than with a wintertime release. This has to do with the Davidson phase of the California current, where there tends to be less cross-shore transport and more near-shore retention. All right, so that's the physics only. Of course, organisms exhibit all sorts of behavior, as we talked about earlier in the talk. There are lots of different behaviors, and this is work that I collaborated with Stephen Morgan about. Stephen identified a number of different organisms that exhibit different behavior in the Bodega Bay area, and many of them are different types of crabs, but barnacles as well. We have in-surface boundary layer. So those are organisms that stay up in the upper 10 meters or so, maybe 15 meters. We've got things which stay below the surface boundary layer. They might be down at 20 meters or 30 meters depth. Diel vertical migration, ontogenetic migration, where they change their depth level at different life stages. Reverse ontogenetic, migration, so things might start up near the surface rather than at depth and go deeper in different life stages, or the reverse. They might start at depth and go shallower at different life stages, and then some combination of ontogenetic and diel vertical migration. So we explored the impact of this biology on the circulation, and then we thought it might have a really big effect because the dominant circulation pattern on the central California coast is the upwelling circulation, which of course winds blow equatorward, and they drive upwelling near shore, and that of course leads to the cold temperatures near shore and high standing stock of chlorophyll. This is actually work that I was involved with many years ago when I was a postdoc, and this is work where Hal Batchelder had developed an individual-based model for copepods. These were calinus copepods, and they grew through different life stages. They actually fed on a biogeochemical model, so we had representation of phytoplankton in this model that's not shown here. But the point is that if you have a diel vertical migration, it can significantly change the nearshore connectivity or the nearshore population that you get, because with no behavior, organisms that are nearshore end up being swept offshore in the nearshore Ekman transport. Whereas, and you can see that here, at 20 days, the copepods, which have large circles, those have grown because that's where their food resources are. But over time, they are not retained near shore. Whereas ones that exhibit a diel vertical migration behavior sample different parts of this cross-shore pattern, and you end up getting copepods that are abundant and thriving near shore. Is that a question, or are you okay? Okay, so as in a three-dimensional circulation structure, you get the same sort of behavior. I'm showing all sorts of behavior on the upper left here. The passive one with no behaviors is this upper right panel, but things that stay below the surface boundary layer end up having a large concentration near shore. There are different patterns. Ontogenetic has a pretty high near shore concentration. The worst are the in-surface boundary layer organisms because they just get swept out to sea. And we can quantify this in some meaningful way by comparing to the passive and looking at the total settlement strength. And you can see that if we look at the total number of organisms that are retained near shore and compare it to the passive one, you can get orders of magnitude more near shore retention through different organismal behavior. Yeah?
Why do you think when you did the three-dimensional model of the passive and the diel vertical migration looks so similar here, right? Whereas they were very different in that 2D model results you showed earlier.
That's a fantastic question. And I'm going to say, I think actually it was a surprise to us when we did this, but I think the three-dimensional circulation pattern had so much heterogeneity in the onshore and offshore flows. So they got, yeah, that you end up getting these filaments which have very large cross-shore velocities, which was not present in the two-dimensional circulation pattern. So that while they were sampling, they tended to sample more high-velocity offshore transport at the surface, which moved them on average more offshore than the onshore transport, which was more diffuse with smaller amplitude. Does that make sense?
It does, yeah, it's complicated.
It is. It has, yeah, so the heterogeneous environment, I think, was the cause of that. And this behavior of having large onshore transport was, they didn't sample that surface heterogeneous behavior if they just stayed below the surface boundary layer. All right, so if you add this organismal behavior, you get all sorts of different connectivity matrices. And so here I just like to emphasize really the differences. The total settlement strength is very large for organisms that stay below the surface mixed layer. If you have things near the surface, they get swept offshore. But all is not lost. There are things which transport far from their origin. It depends a little bit on where they start. Things from Southern California still have very large connectivity up into the southern Central California region. And Monterey Bay still remains as a hotspot of connectivity. When we had DVM, even though there was an overall loss relative to the below case, you can see a lot of these hotspots at particular locations. And with the ontogenetic and reverse ontogenetic, I would say the same variability is true. The ontogenetic showed behavior kind of similar to the below, and then the reverse ontogenetic had more variability with particular hotspots along the coast. All right, so I'd like to move off of connectivity matrices now and talk about how we can learn something about the ocean circulation from these trajectories. This is work, it was a slightly different model domain than what I was talking about before. It was a nested model going down to the Central California coast. You can see the domain of the model on the right. And what I'm showing here is a representation of the Eulerian eddy kinetic energy. So this EKE represents an average, it's an average of the speed of the currents once the climatology is removed. So you have some mean circulation, you have some mean transport, and if you remove that, instead look at the variability, this picture on the right represents the average variability that we had. And what I'd like to point out is that the eddy speeds were large offshore. Sorry, I can't find my mouse. They were fairly large offshore, whereas near shore, you get these regions of fairly low eddy speed. So Monterey Bay, Gulf of the Farallones, even up off of Bodega, we go from blue basically to red. And that heterogeneous nature of the ocean circulation is going to have an impact on the transport or dispersal of organisms. So this is just a representation of Fickian diffusion up on the upper left. This is from the Wikipedia page. If you start with a membrane that divides something with some concentration on the left and no concentration on the right, and then you remove the membrane, because of Brownian motion, things will move from the area of high concentration to low concentration. And so we should expect dispersal to do that in the ocean as well. And what I'm showing on the right are clouds of particles. So these represent those Lagrangian PDFs, kind of the, well, kind of what's shown in the bottom here, for this Central California coast domain after release. So we release them in our entire domain, but then reprocess them and just look at their deviation from their point of origin. And after 4 days, you can see that there's a large alongshore spread and a fairly small cross-shore spread. But over time, that cloud disperses. It moves further in the alongshore direction and further in the cross-shore direction. And after 12 days, you can see it spreads more. After 16 days, it spreads even more. And this little star that's located here, that represents the central moment of all of those, of that Lagrangian PDF. So this represents the average position of that cloud. Anyway, we can develop a theory for this cross-shore dispersion by looking at this cloud. And this is the only math that I'm going to have in the talk, but I thought it might be useful for and of interest to some. So you start with a one-dimensional diffusion equation where y represents the offshore direction. And we assume some sort of linear diffusivity. So we think that dispersion is happening with some constant nu times the distance in the cross-shore direction. If you insert this into this one-dimensional diffusion equation, you can come up with an analytical solution for the concentration of whatever psi is. Could be concentration of particles, for example, shown here. And if we assume that the initial release location is set at a point 0, then this more complicated function with a modified Bessel function ends up having a simple solution that's just an exponential decay. And you can use that solution to develop a representation for the first and second moments of the distribution, where the first moment just represents the average location of the cloud, and the second moment represents the spread around that cloud. And they're shown over on the right. The first moment is here in red, and it's just shown as the initial location, which is going to be 0, the original point, plus some linear increase with time. So what this is saying is that the mean of the dispersing cloud is going to move offshore with some linear increase in time. And the second moment, the spread about that mean, turns out also for long time periods to be linearly related to time. So anyway, we've got the mean moving cross-shore and the spread about the mean moving cross-shore. Those represent two different predictions that we can test with the model. So that's shown here. We've got in black what that mean position is, the star that I showed initially, over the first 16 days after release. And although there's an initial slow period, you can see that the mean moves cross-shore with roughly a linear increase with a value of this constant of 3.3 centimeters per second. The second moment is shown down here, and it also increases kind of linearly with a value of that constant of 3.4 centimeters per second. So those were kind of in line with each other. And then the question is, well, how do we interpret this constant, 3.3, 3.4? And so now I'd just like to explain what this dispersal means. We used what's called a mixing length theory, where we represent the mixing coefficient as a velocity times a length scale. Dimensionally, that's going to be true because velocity is like meter per second, and then length is meters, so you end up getting meters squared per second squared, which is the units of dispersal. And then you just have to interpret what's the velocity and what's the length scale. So originally we thought we'll just let it be this coefficient times the distance y. But if we do that and we calculate a velocity scale from the model, we end up with a value of about 10 centimeters per second, or about 3 times the value of nu that we calculated. So an alternate way of thinking about this is if we think about the dispersal coefficient, the diffusion coefficient, as the RMS velocity times the Rossby deformation radius, where that's given by NH on F, it's largely controlled by the bottom depth. So if we think about the length scale not as the distance from shore, which is kind of a common scale when people do law of the wall type diffusion problems, if we don't think about it as that distance, but instead think of it as a length scale that's associated with the first baroclinic mode Rossby radius, then we get a pretty good agreement between the model and the data. Yeah?
So is that Coriolis?
That F is the Coriolis parameter, and N is the stratification.
So it's a long enough scale though, at that Rossby is relevant?
The Coriolis parameter, well, this parameter it represents a length scale that eddies take at any location in a rotating fluid. And it depends on the stratification and on the bottom depth and on the Coriolis parameter. But anyway, it kind of represents an eddy scale, and in very shallow water that scale shrinks, and then in open ocean water it reaches a magnitude of about 20 or 30 at mid-latitudes here. All right. I'd like to talk about the next topic, which is predictability. So this work was motivated a little bit by work that Dave Siegel was doing down at Santa Barbara with Satoshi Mitarai. There was this really interesting paper where they looked at the variability in settlement associated with the variability in filaments and eddies, and argued that the predictability is very low because of the high variability in ocean circulation. From an observational points of view, there is a lot of evidence that settlement is predictable, and there's been a lot of work on its predictability based on wind and temperature. So we thought we would investigate that. And this is the same model over on the left. And then what I'm showing here is the alongshore wind stress and the surface temperature, and then some settlement from our model. And what you can see is that there's clearly a climatological pattern in all of these structures. You can see that this is for different behaviors, but even with different behaviors, you see some variability with the settlement. And so it seems to be true on climatological scales, but you can even look on instantaneous scales. So over on the left is the climatological scale. I'll skip that for now. But if we look on instantaneous scales, the wind stress shows a lot of variability as it goes back and forth, whereas the settlement, this is for the diel vertical migration case, you can see it kind of tracks the wind, but it doesn't show the same sort of variability. But you can imagine that if you average the wind stress over a long period of time, a lot of the variability is going to drop out. And you can show that if you average it over the pelagic larval duration, you end up getting a very good relationship between the wind stress and the settlement. So we can do this more quantitatively. We can calculate, for example, correlation coefficients with different lags between settlement that we calculate in our model and the applied wind stress averaged over some region. That's shown as this black line, and we find the highest correlation happening at about a lag of about half the pelagic larval duration. Temperature, on the other hand, has a lag of only 2 days in this case, kind of the competency window in this particular study, half the competency window. It turned out that anomalous temperature had higher correlations than absolute temperature, and anomalous wind stress was less correlated with settlement than absolute wind stress. And we can do this more quantitatively still by looking at different frequency bands where we look at the coherence between wind stress or temperature and settlement. And I'll just say in the box that potential settlement we found along the central California coast is very strongly correlated with wind stress when averaged over a sufficient period of time. So if you have a record and you know the pelagic larval duration and it's long, like a week or longer, 20 days, something like that, there's very strong correlation between settlement and wind stress. And this agrees really strongly with observations that go back to Roughgarden back in the 1980s, where they observed a lot of settlement in relaxation periods. All right, I think the last thing that I'll talk about is the impact of horizontal swimming. And this was a really interesting one to me because I had never thought that horizontal swimming for these organisms could be important. There was work on this that was done by a student here back in the 2000s. I don't know if any of you knew Kashef, okay? So there's this nice paper which documents the swimming speed of organisms. And for rockfish that are released, this sort of order 1 centimeter per second. For settled juveniles, maybe in the tens of centimeters per second. And so to me, they also report that the swimming speed just grows steadily over their development period. So one might think that for larvae that are settling, that are in their pelagic period, they're going at kind of 1 to a few centimeters per second, and that's tiny compared to the ocean circulation speed. So I certainly thought that this would be a negligible effect. So we tested a lot of different horizontal swimming speeds. Oh, and they were cued to know what direction they would swim. They had to swim towards the shore, thinking maybe they would cue on magnetic signals or daylight, sunrise, sunset sort of things, or maybe olfactory cues. And we tried a lot of different settlements, swimming behaviors. Nothing for the first several days, and then they would start to swim, and maybe they would ramp up their swimming at some pace. We never went more than 5 or 7.5 centimeters per second. Most of our tests were kind of 5 centimeters per second or 1 centimeter per second. But it had a really big effect on connectivity. So what I'm showing here on the left is a comparison of no swimming behavior to swimming behavior for a particular scenario. And in all cases, you could see that the nearshore concentrations in these PDFs were enhanced by this active behavior of the fish larvae. And the other thing we found was that it was actually kind of predictable. So we found a linear relationship between the maximum distance swam. So if you just do an integral of over the pelagic larval duration of how far they're going to swim, it didn't really matter whether we started late or started early. It had a noteworthy effect. So no swimming is shown by this circle. And then even 1 centimeter per second starting at day 10 had a non-negligible jump in the settlement strength. That's the integrated settlement. Faster swimming or earlier. This I think was starting on day 30. This one starts on day 10. And so on, with this one on the right being 5 centimeters at day 10. With the largest integral. But anyway, it was a surprise to us that even small swimming speeds, like 1 centimeter per second, could have this huge impact. Directional swimming. We tested also if you had some random variability about that. Maybe they couldn't always go to the northeast. It didn't actually matter as long as their variability was random. If it was biased, it would have an effect. But anyway, this was a surprise to me that even very small swimming behaviors could have a pretty big impact. All right, time is getting short, and so I'm going to skip a discussion about fronts in Carmel Bay, even though this is a really awesome movie and study. But I'll just jump to the conclusions. We've been involved in a lot of these dispersal studies for the California current. They're motivated by a lot of different topics. Lately, we've been really interested in marine protected areas and trying to understand how they're working together to support one another. We've found that connectivity along the California coast depends on things like the pelagic larval duration and the season of release, so what currents they're sampling. The behavior also shows sensitivity to, the connectivity shows sensitivity to the behavior. Having said that, there are some real trends that emerge. There are some time mean flows that influence directional transport overall. And there are some places like Monterey Bay which end up being hotspots of settlement, or the Gulf of the Farallones, across many different behaviors. We developed some theory for cross-shore transport, and we looked at the predictability with the wind stress, found it to be quite predictable, highly coherent with the wind stress at long time scales. And then I finished off just with this discussion about how horizontal swimming can increase the nearshore retention and potential settlement by many factors, an order of magnitude, depending on the behavior. All right, well, thank you very much, and it was a real pleasure talking to you.
questions, I'm gonna bring the lights up. I had one right off the bat. I'm thinking, I mean, one of the great things about models, right, is as we get better and better at them, we get more and more detail, and we get to visualize features better. For a lot of us, it just helps us put things together. The second part, which I think where you guys are, is the ability to forecast. And so as we think about all the predicted changes, especially in the longshore wind stress that are expected for the California current, have you played that game? And specifically, let me just catch it one way, specifically in the changes in connectivity of the marine protected areas under future.
Yeah, great question. We have submitted a few proposals to do that, and we continue to do so. And I have a feeling that ultimately we will be able to answer that. One of the things that collaborators that I work with do is they do predictions out to 2100. This is work that Mariposa Wheel and Jerome Fischer do. And we would like to understand what the impact is on the dispersal. There are going to be changes to the California current. Certainly temperatures are going to change. Those are quite robust predictions. I think the changes to the dispersal are not as obviously predicted. So one of the things that will change likely is the eddy kinetic energy, and that is this, it'll change because the stratification is going to change, and that's going to change this Rossby deformation radius, and it's also going to change because the wind stress may change in the longshore direction, and that'll change the energetics of these things. I'm not going to make a prediction without knowing what the model really was going to say. So I'm going to say super interesting area. We'd like to know how this is going to change as well, but we haven't done the study yet. Yeah.
Have you been able to like look back at like the blob event and see how that has changed?
Oh,
You know,
interesting.
circulation and connectivity patterns. It's kind of an analogous study on what may happen further in the future.
Yeah, so all of the work that I've been talking about has not been event-driven. It's been more climatological averages over many, many years. And the blob is a really interesting one because there was reduced upwelling that year, there was, or those years, there was reduced eddy kinetic energy, and there should be impacts of dispersal. I don't have an answer for that one either, sorry. We've mostly focused on these climatological averages, but the intraannual variability is super interesting. Thank you for asking.
For the horizontal swimming, I was curious. I know you mentioned that like it's such a small amount compared to the journey that they have to take in order to get to shore. You ever been to the Saws and Settle? So could it be less that like that little itty bitty amount that they're swimming is getting them there, and more that those that swim for some other reasons, maybe because they can forage, or maybe because they can, I don't know, do other stuff that goes with swimming that allows them to survive long enough to get there?
So in nature, absolutely. So in nature, there are going to be all sorts of biological factors, which I've neglected because it's complex enough as it is, and I really wanted to just focus on the physical effect. Almost certainly, if they're moving, particularly vertically and also potentially horizontally, they're moving into different food resource environments, and they're also moving potentially into different predation environments. That's going to have an impact on their survival, for sure.
And when you talked about it, like for the horizontal swimming, I guess what was happening with that you talked about? It wasn't in nature. It was.
So I examined if organisms could control their horizontal position by actively swimming in a particular direction. So we had them swim towards the shore at a speed which was relatively modest compared to the amplitude of the currents that they experienced, and that tiny little bias in their swimming ended up having a quantitatively important effect on their ability to stay where we think their best habitat is. When I was a postdoc, I remember someone made the point that the average larva is dead, which is a terribly depressing statement, but it is true, right? So the very large number of organisms that are released. They get released into the water column. Most are going to die for a number of reasons. They get transported to an area with low food content. They get eaten along their way. It's very few which make it back to their habitats where they can themselves settle and spawn. And so, anyway, we're just steadily trying to explore how different life strategies can impact that. Yeah.
On the subject of different life strategies, I like the looking at wind stress limits, trying to figure out like out of all the factors which is most important by lag time as well. I thought that was very cool. But also I was wondering that how does wind stress affect the different larval strategies of like staying in the surface versus like 30 meters down?
Are you asking a biological question about whether they sense the turbulence that takes place in the upper water column and then move actively for that? Are you asking a biological question or a physical question? Because I can answer the physical question. I don't know if I
I
can
might be
answer.
drifting a bit too far into the biological.
Yeah, I'm sorry. I'm going to get myself in trouble if I answer that in some way. I'm sure I'll be wrong. What I have heard is that organisms can, I mean, it depends. When they're very small, they get moved by the turbulence in the upper water column. As they get larger, they develop the ability to resist that movement by the physical transport. And so as they grow, there is this suspicion that they are not moved randomly by the turbulence as much. But it depends a lot on as to which organisms we're thinking about. If we're thinking about, for example, rockfish larvae, they're pretty large. On the other hand, if we're thinking about eggs from Calanus copepods, those are awfully small, even when they're at later life stages. Yeah.
I was curious what software you used to make the animation, and if you could go back to it because it's really pretty.
Oh, thanks. I'm assuming you're going to talk. I'm assuming you're referring to this one,
Yeah.
which is fantastic,
It's
and
awesome.
I totally credit my student Anna Lowe, who spent some time to make beautiful animations for this. So I can say that she did everything in Python. The larval transport calculations were done using an open source piece of software called OpenDrift, which is downloadable. It's a Python based code. It takes the model output and it calculates the solution to dx dt is equal to u, and it prints those trajectories. Now, once those are calculated, it saves the position as a function of time. Once those are calculated, then you need some way to visualize it, and this is done, I think, using Matplotlib, but honestly, I'm not sure what she did.
That's okay.
It is fantastic, though.
It's gorgeous. Thank you.
Sure. Yes.
And so the things you released are called PDFs?
No, we released particles like this, as
Yeah, I
shown
was
in this
curious what they
figure.
actually
but-
look
but-
like.
I'm sorry.
What the actual particles look like that you released.
This is what they look like. Those black dots.
Okay.
So they're literally, they're literally tiny. They have no size. They're just point particles within our numerical model, and they get moved by the currents, and we just keep track of where they are. We calculate the Lagrangian PDFs by running millions of these particles, and then over time, calculating the density of particles that we see in different parts of the domain. So we can calculate, oh, there are 50,000 here, there are 100,000 over here, and then we sample them spatially and create these two-dimensional probability density functions.
I was just curious how you made them at different, in the water column,
Oh, what I showed here were all top-down views. So what I showed here were simply PDFs where we didn't distinguish whether they were in the upper part of the water column or the lower part of the water column, even though they were distributed in the vertical. So we just said, oh, if they happen to be in this latitude, longitude range, we're going to count that as being in that range. So my understanding is that when rockfish larvae, for example, are ready to settle, they may arrive at a particular location in the upper part of the water column, but they'll ultimately sink down towards the bottom for their settlement. So it didn't matter to us what part of the water column they were. We just counted them in that latitude, longitude box.
So now that you're on to some images you've already shown us, you showed us one or two earlier, where the retention centers start to visualize, right? Where, I forget what the graphs were, but you were looking at the evolution of the model, and early it was all red along the 1-1 line, and then, all right, any one of those. Oh, actually,
I'll go to the one with.
I think, yeah, you probably know which one. There we go.
Yeah.
Now you're getting these, so the far one on the right, which is the DVM, you're getting these retention centers, right? The red hotspots, whereas the entire area is not just red.
That's right.
I'm understanding that, right? Yeah,
That's,
so,
that's exactly right.
so my question is, knowing your co-authors, right, who are benthic ecologists, have you overlaid that onto
Oh,
the
habitat
biogeographic patterns and asked, does the retention centers of the model overlay with, you know, for example, these ideas when you're in Washington and you don't have much movement, everything gets retained and you get massive barnacle sets, massive mussel sets, whereas in California, where much of the coast is in between those, you get seaweed, right? Because you don't get these things that must be retained to stay close. This is a very selfish question.
No, it's awesome. So I'm not going to give you an answer I think that satisfies you. So we have looked at habitat effects, where if we take out some of these locations, we just take them out. There's no settlement that takes place in those areas, and there are other regions nearby that may be rocky and available where they do then tend to congregate and create new source regions. And we focused on the marine protected areas, particularly focused on how they're communicating with one another. Here in Monterey Bay, of course, there are large areas sand covered which don't have good rocky habitats. And even though our model would predict organisms in this area, they're not going to settle there. So in this qualitative sense, yes, but the way we think of it is we just remove these from the connectivity matrices. And our co-authors do take these and look at, all right, these are spots that would tend to be useful or are known to be hotspots.
All right. Well, another thank you very much, Chris.